Heredity and Evolution. Now on the first page, name of the scientist here and what they discovered, it is given. First of all, what is the meaning of heredity? Heredity you have studied last year also. It is a transfer of the biological characters from one generation to the next generation with the help of genes or through the genes. Now the first scientist, Johann Gregor Mendel, in the year 1886 was a pioneer of the modern genetics. So whatever we are studying today about genetics, he was a one who originated all this modern genetics. Next scientist, Hugo de Virus, in 1901, he was the one who discovered the mutational theory. Because of the mutational theory, the reasons behind the sudden changes were understood. Walter Sutton in 1902 observed the paired chromosomes in the cells of a grasshopper for the first time. Now you'll be studying all these chromosomes and all. You have studied last year also about chromosomes. So he was the first one to discover the chromosomes. Oswald Avery, three scientists together, Oswald Avery, MacLean McCarthy and Colin MacLeod in 1944, they proved that except viruses, all living organisms have DNA as genetic material. The genetic materials there in each and every living organism, except viruses don't have this DNA of genetic material. Then French genetist Francis Jacob and Jacques Monod in 1961 proposed a model for the process of protein synthesis with the help of DNA in bacterial cell. You know the model of process of protein synthesis, we are double helix strands of the DNA and all. This was possible because of the model proposed process of the protein synthesis was first proposed by whom? French genetist Francis Jacob and Jacques Monod. Now because of this French genetist, it helped to uncover the genetic codes hidden in the DNA. Next you will be studying about this only. Hence the technique of recombinant DNA technology emerged, which has a vast scope in the field of genetic engineering. Now science of hereditary. The science of hereditary is useful in many fields. What are they useful for? Diagnosis, treatment and the prevention of hereditary disorder. You have studied that in last year. What is hereditary disorder? What is hereditary disorders? Like sickle cell anemia and all is hereditary disorder. So because of genetic engineering, it is possible to diagnose this hereditary disorder. Treatment can be possible and prevention of these disorders can also be possible because of genetic engineering. And production of hybrid varieties of plants and animal also was possible because of this. And in industrial process also it is useful, in which microbes are used. This you will be studying in lesson number 7. So that time you will come to know what is the meaning of this. Now this is the DNA structure. There is a question on page number 1. Explain with diagram the structure of DNA and RNA. So first we will discuss about DNA and then we will discuss about RNA. It's very important that you know the structure of DNA, then only you will understand the next topic of this lesson, that is transcription, translation and translocation. Now the DNA structure. What is the full form of DNA? The full form of DNA is what? Deoxyribonucleic acid. Now the DNA structure is made up of two parallel threads. So these are the two parallel threads which are coiled around each other. So it will be coiled around each other this way. And in between these two threads of DNA, there are nitrogenous base between them. So there is nitrogenous base between the two parallel threads which are coiled around each other. Now if you look at this blue color which I have shown in the diagram is the two parallel threads, and in between, which I have shown in black color, is the nitrogenous base. There are 4 types of nitrogenous base. What are they? Cytosine, guanine, adenine, thymine. So these are the 4 nitrogenous base. The cytosine always pairs with guanine and adenine always pairs with thymine. So if this is cytosine over here, then this will be guanine. If this is adenine, this will be thymine. If it is guanine, cytosine, thymine, adenine, cytosine, guanine, adenine, thymine, guanine, cytosine, thymine, adenine, cytosine, guanine, adenine, thymine, guanine, cytosine, thymine, adenine, guanine, cytosine, and adenine and thymine. So adenine always pairs with thymine and cytosine always pairs with guanine. अभी ये डीएनए स्ट्रक्चर है, पेज नंबर 1 में क्वेश्चन है डायग्राम के साथ एक्सप्लेनेशन देने का, डीएनए स्ट्रक्चर और आरएनए स्ट्रक्चर. ये बहुत इंपॉर्टेंट है कि स्ट्रक्चर पढ़ के लेना, ये 9th स्टैंडर्ड में पढ़ा हुआ है. अगर ये समझ में आ गया तो ट्रांसक्रिप्शन, ट्रांसलेशन और ट्रांसलोकेशन समझने में थोड़ा इजी पड़ेगा. नाउ डीएनए स्ट्रक्चर, डीएनए का फुल फॉर्म क्या है? डीऑक्सीराइबो न्यूक्लिक एसिड. डीएनए स्ट्रक्चर 2 पैरेलल थ्रेड से बना हुआ है, तो 2 थ्रेड्स हैं और ये दोनों थ्रेड्स कॉइल्ड अराउंड ईच अदर है. तो दोनों भी थ्रेड ऐसा पैरेलल नहीं है मगर इट इज़ कॉइल्ड अराउंड ईच अदर. और इनके बीच में क्या है? नाइट्रोजनस बेस. तो ये ब्लू कलर जो है वो पैरेलल थ्रेड्स है और बीच में जो ब्लैक कलर में दिखा है वो नाइट्रोजनस बेस है. नाइट्रोजनस बेस 4 टाइप का है, साइटोजाइन, ग्यूनाइन, एडेनाइन और थाइमाइन. साइटोजाइन जो है हमेशा ग्यूनाइन के साथ पैर होता है और एडेनाइन जो रहता है वो हमेशा थाइमाइन के साथ पैर होता है. नाउ एडेनाइन एंड ग्यूनाइन is known as what together? Purines. And cytosine and thymine is known as what together? Pyrimidines. तो एडेनाइन और ग्यूनाइन दोनों को क्या बोलते हैं? प्यूराइंस. और साइटोजाइन और थाइमाइन दोनों को क्या बोलते हैं? पिरिमिडाइन. अभी यहाँ से लेके यहाँ तक का डिस्टेंस कितना है? हाउ मच इज़ द डिस्टेंस फ्रॉम ईयर टिल ईयर? इट इज़ 3.4 nanometer. And the distance from here till the first nitrogenous base is how much? 0.34 nanometer. And the distance between this from here till here is how much? 2 nanometer. Now the 2 parallel threads is somewhat looking like this. It is made up of alternate phosphoric acid and sugar molecule. What is the meaning of that? If this is phosphoric, this is sugar, then phosphoric, then sugar, then phosphoric, then sugar, and so on. So the 2 threads, the parallel threads of the DNA is made up of alternate what? Phosphoric sugar, phosphoric sugar, phosphoric sugar, and so on. And in between the nitrogenous base are made up of what? Cytosine, then this will be guanine, this will be, if this is adenine, this will be thymine, and both the nitrogenous base are joined by hydrogen bond. Hydrogen bond. So this is the phosphoric acid. This is the sugar molecule. So this is the structure of DNA. Now I will discuss the structure of RNA. Now RNA, what is the full form of RNA? Ribonucleic acid. Now the RNA is also made up of phosphate molecule like the DNA and sugar molecule, and it has 4 types of nitrogenous bases. What are they? Cytosine, guanine, adenine, but instead of thymine it has uracil. So RNA does not have thymine, it has uracil. So full form of RNA का full form क्या है? राइबोन्यूक्लिक एसिड. वो भी DNA के जैसे फॉस्फेट मॉलिक्यूल और शुगर मॉलिक्यूल से बना है और 4 नाइट्रोजनस बेस है, साइटोजाइन, ग्यूनाइन, एडेनाइन. लेकिन DNA में थाइमाइन है और RNA में यूरासिल है. There are 3 types of RNA. What are they? rRNA, mRNA and tRNA. The first one is rRNA, since it is the component of ribosome, therefore it is known as ribosomal RNA. What does it does? It does the function of, or it performs the function of protein synthesis. Now the second type of RNA is mRNA. Now mRNA is known as messenger RNA. It is known as messenger RNA. It carries the information from a gene, that is DNA. Gene is also known as DNA, that is the DNA in the nucleus up to the ribosome in the cytoplasm. Ribosome in the cytoplasm. And it produces what? Produces protein. So I'll repeat once more. It is known as messenger RNA. It carries the information of the gene, which is DNA. That is a DNA which is inside the nucleus. So it carries the information from the gene or the DNA in the nucleus up to the cytoplasm. So this is the cytoplasm. So up to the cytoplasm, up to the ribosome, which is in the cytoplasm. So up to the ribosome, which is in the cytoplasm. So it carries the information from the DNA up to the ribosome, which is in the cytoplasm, and it produces proteins. The last one is tRNA, or the transfer RNA. So this is also known as transfer RNA. Now what does this do? According to the message of mRNA. So according to the message, whatever message mRNA has brought, so to the message of mRNA, it carries amino acid. It carries amino acid to ribosomes in cytoplasm. So there are 3 types, 3 टाइप ऑफ RNA है. एक तो है फर्स्ट वाला है rRNA. इसको क्या, एक कंपोनेंट ऑफ राइबोसोम, इसके लिए राइबोसोमल RNA इसको बोलता है. इसका फंक्शन क्या है? प्रोटीन सिंथेसिस. सेकेंड टाइप है मैसेंजर RNA. मैसेंजर RNA क्या करता है? इट कैरीज द इंफॉर्मेशन. कहाँ से कैरी करता है? DNA से कैरी करता है. DNA किधर है? न्यूक्लियस के अंदर. तो DNA न्यूक्लियस के अंदर रहता है और इन्फॉर्मेशन जो डीएनए दिया रहता है वो इन्फॉर्मेशन न्यूक्लियस से लेके राइबोसोम में लेके जाता है। राइबोसोम किधर है? साइटोप्लाज्म के अंदर है। तो यहाँ पे साइटोप्लाज्म है, तो ये डीएनए इन्फॉर्मेशन राइबोसोम के पास लेके जाता है विच इज़ इन द साइटोप्लाज्म, और ये क्या प्रोड्यूस करता है? प्रोटीन्स। लास्ट वन टीआरएनए, इसको ट्रांसफ़र आरएनए बोलता है, और इसका काम क्या है? अकॉर्डिंग जो भी मैसेज एमआरएनए लाया है उसके अकॉर्डिंग से वो अमीनो एसिड कैरी करता है राइबोसोम टू राइबोसोम इन द साइटोप्लाज्म। सो दिस इज द डिफरेंट टाइप्स ऑफ आरएनए। Of central dogma or transcription, translocation and translation. What is the meaning of that? I'll explain that. Now DNA is a double helix which we have already studied last year. So it's in the form of double helix and it is double stranded, and in each strand there are nucleotide. Now if I draw this spiral form of DNA in this form, in straight form, then it will look like this, and these are the nucleotide in it, and so on. Supposing if this is C, G, A, T, T, A, and so on. What is C, G? C is cytosine, G is guanine, A is adenine, T is thymine, and so on. Now we know that cytosine always pairs with guanine and adenine always pairs with thymine. So if this is cytosine, this will be guanine. If this is guanine, this will be cytosine. If this is adenine, this will be thymine. If this is thymine, this will be adenine. Thymine, adenine, and adenine, thymine, and so on. अभी लास्ट ईयर अपन पढ़ा था कि डीएनए स्ट्रक्चर कैसा रहता है? डबल हेलिक्स फॉर्म में रहता है, स्पाइरल फॉर्म में रहता है, और हर एक स्ट्रैंड ऑफ डीएनए में क्या रहता है? न्यूक्लियोटाइड रहता है। और न्यूक्लियोटाइड कौन सा रहता है? साइटोसाइन, ग्यूनाइन, एडेनाइन और थाइमाइन। अगर मैंने यही डबल हेलिक्स फॉर्म, स्पाइरल फॉर्म को ऐसा सीधा बनाया तो कुछ ऐसा दिखाई देगा। अगर समझो ये सब न्यूक्लियोटाइड, समझो साइटोकाइन है, साइटोसाइन है, ग्यूनाइन है, एडेनाइन है, थाइमाइन, एडेनाइन, कुछ भी हो सकता है। अभी साइटोसाइन हमेशा ग्यूनाइन के साथ ही पैर होता है और एडेनाइन हमेशा थाइमाइन के साथ ही पैर होता है। इसके लिए अगर ये साइटोसाइन है तो ये ग्यूनाइन है, ये ग्यूनाइन है तो ये साइटोसाइन, ये एडेनाइन है तो ये थाइमाइन, अगर ये थाइमाइन है तो एडेनाइन, थाइमाइन, एडेनाइन, एडेनाइन, थाइमाइन, एंड सो ऑन। अभी इसका कॉपी, डीएनए का कॉपी बनता है, और ये डीएनए का कॉपी जो बनता है उसको बोलता है एमआरएनए। डीएनए में 2 स्ट्रैंड है मगर एक ही स्ट्रैंड एमआरएनए बनने में यूज़ होता है। सो इन ऑर्डर टू मेक एमआरएनए दे मेक एग्जैक्ट कॉपी ऑफ डीएनए. A DNA copy is made which is known as mRNA, and in forming mRNA only there is double strand in DNA, only one strand is used for making mRNA. If this is guanine then this will be cytosine, if this is cytosine this will be guanine, if this is thymine this will be adenine. If this is adenine, in mRNA you have studied last year there is no thymine, instead of thymine there is what? Uracil. If this is adenine, uracil, thymine, adenine, and so on. And so on. So the making of mRNA from a DNA is known as what? Transcription. So this is known as transcription. Now the only difference between DNA and RNA is what? DNA is double strand but RNA is a single strand. In DNA thymine is there, in RNA uracil is there. अभी DNA से कॉपी बनता है RNA. अगर ये guanine है तो ये cytosine, ये cytosine है तो ये guanine, thymine है तो ये adenine, और adenine है तो ये uracil, adenine, uracil, और thymine, adenine. अब ये mRNA जो बनता है, ये प्रोसेस जो mRNA बनता है उसको बोलता है ट्रांसक्रिप्शन. नाउ दीज codes which are there in mRNA is in a coded form. So this code has to be translated. So who translates this code? tRNA translates this code. So it is translated by tRNA. The tRNA has the anticodes of these codes. So if this is C, G, A, UUA. So the codes of these are in coded form. The anticodes of this code is there in tRNA, and the tRNA which translates this codes of mRNA is known as what? Translation. So this is known as translation. Now these anticodes which bring the amino acid, this is the amino acids over here, this is the amino acid. They bring the message of which nucleotide? Only 3 nucleotides. They bring the message. You can see over here 1, 2, 3. So it brings the message of only 3 nucleotides, and each tRNA brings 1 amino acid. The next 3 anticodes of this mRNA is brought by the next tRNA, which brings another amino acid. So this 3 codes which is brought, which is translated by the tRNA is known as triplet codon. अभी mRNA जो है उसका जो न्यूक्लियोटाइड है, जो न्यूक्लियोटाइड है वो सब कोडेड फॉर्म में रहता है, और इसका कोड का मैसेज किसके पास है? tRNA के पास है. tRNA जो है उसके पास एंटी कोड है जो इसको ट्रांसलेट करेगा और अमीनो एसिड जो रिक्वायर्ड है वो लेके आता है. सो इफ दिस tRNA ब्रिंग्स द कोड ऑफ दिस 3, तो ये 3 का कोड लेके आता है, ये 3 का लेके आता है, इसके लिए इसको क्या बोलता है? ट्रिपलेट कोडॉन. तो इसको क्या बोलता है? ट्रिपलेट कोडॉन. क्यों ट्रिपलेट कोडॉन बोलता है? क्योंकि हर tRNA सिर्फ 3 न्यूक्लियोटाइड का मैसेज लेके आता है और अकॉर्डिंग टू दैट कोड अमीनो एसिड लेके आता है. सो दिस इज द अमीनो एसिड विच इज ब्रॉट. द नेक्स्ट one is the next triplet codon, brings another amino acid over here. Now this formation, this is translating by the tRNA the codes of mRNA is known as translation. तो जो एंटी कोड्स जो tRNA लेके आता है, जो ट्रांसलेट करता है कोड ऑफ mRNA, उसको बोलता है ट्रांसलेशन. सो यू अंडरस्टूड व्हाट इज ट्रांसक्रिप्शन एंड ट्रांसलेशन. अभी जो सेल रहता है उसके अंदर न्यूक्लियस रहता है. न्यूक्लियस के अंदर, इनसाइड द न्यूक्लियस यू विल फाइंड द DNA. Now this DNA, with the help of this DNA, mRNA is formed in the nucleus. So जो mRNA है वो न्यूक्लियस के अंदर बनता है. क्यों न्यूक्लियस के अंदर बनता है? क्योंकि न्यूक्लियस के अंदर DNA रहता है. Then through the pores, of the nucleus में pores रहता है. Through this pores the mRNA comes into the cytoplasm. So it comes into the cytoplasm, the mRNA, and in the nucleus, outside the nucleus there are ribosomes, not ribosome, endoplasmic reticulum. Now this endoplasmic reticulum has ribosomes in it, and this ribosomes goes from one end of the mRNA to the other end. So the ribosome, when the ribosomes goes from one end to the other end, it is known as what? So when the ribosome goes from one end of mRNA to the other end, through the other end at a distance of triplet codon. So when the ribosome goes from one end to the other end, it passes through triplet codon. One triplet codon and forms amino acid, then the next triplet codon and forms a amino acid. Now these amino acids, so going from one end to the other end of mRNA is known as what? Is called translocation. जो mRNA है वो न्यूक्लियस में बनता है. न्यूक्लियस से mRNA निकल के साइटोप्लाज्म में आता है। जब साइटोप्लाज्म में आता है तो इधर एंडोप्लाज्मिक रेटिकुलम है न्यूक्लियस के बाजू में। इस एंडोप्लाज्मिक रेटिकुलम में राइबोसोम रहता है और ये राइबोसोम एक एंड ऑफ mRNA से लेके दूसरा एंड में जाता है। लेकिन जब भी वो एक एंड से दूसरा एंड में जाता है तो एक एक ट्रिपलेट कोडॉन साथ में, मतलब एक बार एक ट्रिपलेट कोडॉन लेता है, फिर नेक्स्ट ट्रिपलेट। तो जब भी राइबोसोम्स एक एंड से दूसरा एंड में जाता है तो उस प्रोसेस को क्या बोलता है? ट्रांसलोकेशन। सो व्हाट इज ट्रांसक्रिप्शन? द मेकिंग ऑफ mRNA or copying of mRNA from the DNA is known as transcription. What is translation? The codes which are given by mRNA is translated by tRNA because it is having the anticodes of codes of 3 nucleotides of mRNA and brings one amino acid. Now what is translocation? What is the meaning of translocation? Translocation is when the ribosome goes from one end of the mRNA to the other end through the distance of triplet codon is known as translocation. Now this full process, now ये amino acid, this amino acid which is formed over here, like example of one amino acid, it's given in the textbook, that is lysine. So many amino acid is brought by each triplet, brought by tRNA, which is joined by peptide bonds. Peptide bonds, they are joined to each other by peptide bonds. So if many amino acid, chains of amino acids together form what? Proteins. And these proteins are very important for what? Functions of our body. All the functions are done with the help of the protein. So protein helps in the functions of the living organism. As well as the appearance also. In appearance also it helps. Like for example, if a person is blue-eyed, so the blue-eyed the code of the blue-eyed should be there in mRNA, and when the tRNA brings the anticode of the blue-eyed, so our eyes become blue color. So the appearance and the functions of the living organism is done by the proteins, and this protein is done with the help of many amino acids. This amino acid is brought by the tRNA. The tRNA brings the anticodes of the amino acid according to the codes of the nucleotide present in the mRNA. The mRNA is formed in the nucleus with the help of DNA. It makes a copy of DNA and forms the mRNA. Forming of mRNA is known as transcription. Translating the code of the mRNA with the help of the tRNA is known as translation. And when the ribosome goes from one end of the mRNA to the other end through a distance of triplet codon is known as what? Translocation. And this amino acid helps in the process of making proteins. So many amino acid forms complex proteins. What is the use of complex protein? They help in the functioning. So all our body function is done with the help of this protein synthesis. So from the DNA, the formation of mRNA up to the protein synthesis together is known as central dogma. Now Dr. Hargovind Khurana was an Indian scientist. So an Indian scientist whose name was what? Dr. Hargovind Khurana. Had discovered what? Discovered the triplet codons of 20 amino acids. Now you know what is the meaning of triplet codon. So each triplet codon has a message. It brings a particular amino acid. So code of a particular codes of the mRNA brings a particular amino acid, and he was successful enough to find out the triplet codons of 20 amino acid. For this he was awarded Nobel Prize in the year 1968 along with 2 other scientists. Now in page number 2 it is given at the bottom living organism can produce new individuals. Supposing a dog is there, let us take a particular species dog. It gives birth to puppies. So let us consider it gives rise to 2 puppy, and both the puppy don't look similar to each other. So living organism can produce new individual like themselves. So dog give birth to a dog through reproduction, and some of the characters are transmitted to the next generation, which is very similar to the parent. So some characters of the puppies look very similar to the parents because some genes are transmitted to the next generation without any changes. Due to this, some of the characters of parents are transmitted to their offspring. Sometimes sudden changes occur in the genes. So we have studied while copying or while making the mRNA, while copying the DNA, some errors may take place in this, or sometimes a part of the DNA may get altered. Because of this, mutation occurs. Now these mutation can be harmful or can be harmless. When some mutation may be minor, which is harmless, like supposing the parents are both black-eyed but the child will be having blue-eyed, so that is minor mutation and which is harmless. But some mutation may be considerable, means may be harmful also, which we had studied last year. And this, for example, sickle cell anemia, which is a harmful mutation.